I want you to consider just for a moment the actual physiology of a panic attack.
Oh, yeah. It's intense.
Right, like you're sitting completely still,
Yeah.
perhaps just reading an email, and then suddenly your visual field narrows.
Your heart just starts hammering against your ribs.
Exactly. Your respiratory rate spikes, you get that cold sweat breaking out across your skin, and you feel this overwhelming visceral sense of impending doom.
Which is terrifying, obviously.
Massively terrifying. And as clinicians, we often categorize this experience under the umbrella of a psychiatric disorder, right? Like a psychological event.
Yeah, we treat it as a mind problem.
Right. But the terror you feel in that moment isn't merely, a cognitive abstraction. It is a material reality.
Absolutely.
It's a devastatingly rapid cascade of physical molecules just flooding your bloodstream, binding to receptors, and fundamentally altering your brain's architecture in real time.
It really forces a profound reevaluation of how we approach psychiatric illness, doesn't it? I mean, the hardware of the body and the software of the mind, they're not running on parallel separate tracks.
Right.
They are inextricably bound. And this is the realm of psychoneuroendocrinology. It's a field that demands we just, completely discard the artificial boundary between the physiological and the psychological.
Spot on. And today we are doing a deep dive into the mechanisms that govern that interface. We're drawing from Chapter 1.12 of Kaplan and Sadok's comprehensive textbook of psychiatry.
A brilliant chapter, yeah.
It really is. And our objective today isn't just to, list off hormones. We want to understand how internal physiological states actually shape our emotional experience.
Which is the core of our work.
Exactly. But to frame this journey, we have to anchor ourselves to a foundational definition. And that is, psychiatry is the study of how brain, mind, and experience interact, and critically, how that continuous process can go wrong.
Yeah, that definition is absolutely vital because the endocrine system, well, it acts as the primary translator in that interaction.
The translator, I like that.
Right, because how does an external social stressor become an internal state of cellular inflammation? Or, how does a genetic predisposition become a full-blown depressive episode under the weight of trauma?
Right.
The answer is always found in those chemical messengers crossing the blood-brain barrier.
Yeah, and, I was struck by how the source material challenges our classical analogies here, because I used to visualize the brain and the body as a sort of master and servant dynamic.
I think we all did, yeah.
Right. The brain sits up there in the skull, the CEO of the organism, sending down orders to the adrenal glands or the thyroid to just, execute commands.
The old top-down model.
Exactly. But reading this chapter, it feels so much more like two authors co-writing a novel in a shared real-time Google Doc.
Oh, that's a great way to put it.
Right. The brain types out a perception, say. It registers a physical threat. But then the endocrine system immediately jumps in and edits the very next paragraph by releasing a surge of hormones.
And then the brain has to read that edited paragraph, right? Which alters its own inspiration for the following chapter.
Exactly.
It's just this continuous, instantaneous feedback loop. I mean, the classical model of the endocrine system, what we might call the postal service model, was highly linear.
The postal service model.
Yeah, we believed a discrete gland, like, say, the pituitary, wrote a chemical letter, dropped it in the bloodstream, and it traveled to some distant target organ to be opened and read.
A very slow, unidirectional delivery system.
Precisely. But advances in neuroscience have just completely dismantled that simplicity. The brain isn't just a passive recipient of these hormonal letters.
Right.
The central nervous system itself is a massive secretory organ. I mean, neurons synthesize and release peptides that act as hormones.
Which is wild to think about.
It is. And furthermore, the brain is an incredibly eager reader. It serves as a primary target organ for endocrine action. So the distinction between a classical neuron and an endocrine cell has become, well, entirely dependent on the specific physiological context and the microenvironment.
Right, and for the clinician listening, this means that when a patient presents with profound fatigue or anhedonia or sudden onset anxiety, we cannot limit our differential to purely software issues.
No, not at all.
The biochemical state of the body is actively co-authoring that psychiatric presentation. But to understand how that co-authorship actually works, we need to examine the structural rules of these molecules, right?
Yeah, we had to look at the chemistry.
So the text divides hormones into two broad structural classes, and that structure completely dictates their clinical behavior.
Form dictates function. It always does.
Right.
So broadly, we have the protein, polypeptide, and glycoprotein hormones on one side and the steroids on the other.
Okay, let's break those down.
Right, so the protein-based hormones, such as adrenocorticotropic hormone, or ACTH,
Hmm.
and the endogenous opioids like beta-endorphin, these are large hydrophilic molecules.
Hydrophilic, so water soluble.
Exactly. And because they're water-soluble, they cannot easily diffuse across the lipid bilayer of a cell membrane.
Oil and water, they don't mix.
Right. So therefore, the cell has to synthesize them in advance and pack them into these little intracellular vesicles where they just sit and wait for an external trigger to be released.
They need a highly coordinated exocytotic event to get out.
Spot on.
Yeah.
And once they're released into the extracellular fluid or the bloodstream, they travel to their target and bind to receptors located right on the external surface of the target cell membrane.
Like G-protein coupled receptors.
Exactly. And that initiates a secondary messenger cascade inside the cell.
Okay, so that's the proteins. But the steroids, they operate on a fundamentally different paradigm, right? We're talking about cortisol, estrogen, testosterone, and progesterone.
Yeah, steroids are lipophilic. They are derived from a common precursor, which is cholesterol.
Right.
And because they are lipid-soluble, you cannot trap them in a lipid-bound vesicle. I mean, they would simply slip right through the vesicle wall.
Like a ghost through a wall.
Exactly. Consequently, steroids are synthesized de novo on demand. They aren't stored up.
Oh, wow.
Yeah. Once synthesized, they diffuse freely out of the parent cell, travel through the bloodstream. Usually they have to be bound to specific carrier proteins like cortisol-binding globulin, because blood is mostly water, and then they diffuse directly through the lipid membrane of the target cell.
So they bypass the surface entirely. They don't need a receptor on the outside.
Exactly. They just hunt for receptors in the cytoplasm or directly inside the nucleus.
That's fascinating. And this difference in solubility, it explains the timing of different physiological responses, doesn't it?
It does, yeah.
Because a peptide hormone can be released instantly from its vesicle, whereas a steroid response might require, the time to actually synthesize the molecule from cholesterol.
Right, there's a lag. And beyond their structure, the text outlines their functional reach. So we have autocrine action, which is acting on the secreting cell itself, paracrine action, which is acting on neighboring cells, and then your classical endocrine action via the bloodstream.
Right.
We also see these molecules acting as neuromodulators, altering the synaptic environment to make a neuron more or less responsive to classical neurotransmitters like dopamine or serotonin.
Right, setting the tone. And in tracing the evolutionary history of these molecules, we encounter this principle of pleiotropy, which I think is absolutely essential for understanding psychiatric side effects.
Pleiotropy is biological pragmatism at its finest. I mean, evolution rarely invents entirely new molecules for new physiological challenges.
Right, it's too expensive.
Exactly. Instead, it takes an ancient peptide that evolved in a single-celled organism hundreds of millions of years ago and repurposes it to serve wildly different functions in a complex mammalian system.
It's like the ultimate recycling program. So a single hormone might bind to a membrane receptor in the liver to manage glycogen. Yeah.
And then a cytoplasmic receptor in the immune system to suppress inflammation.
Right.
And then a nuclear receptor in the hippocampus to consolidate memory. All the same hormone.
Exactly. This plasticity allows an organism to adapt holistically to environmental demands.
But, practically speaking, for a clinician listening to this, pleiotropy sounds like an absolute nightmare.
Oh, it really does.
If a single hormone, like estrogen, exerts a dozen different effects depending on where it lands, how is the body not constantly falling into systemic chaos?
Very fair question.
Furthermore, if we want to treat a psychiatric symptom using a hormonal intervention, how on earth do we avoid collateral damage across the entire physiological system?
Well, that complication has been the major hurdle in endocrine pharmacology for decades. But the chaos is prevented by an elegant biological mechanism known as tissue specificity.
Tissue specificity, okay. Yeah. When a steroid hormone enters a target cell and binds to its receptor, that initial binding is only the very first step. The hormone receptor complex must then recruit a specific cluster of coactivators and copressors before it can actually alter gene transcription.
So the hormone is essentially a master key, but the lock requires several other highly specific tumblers to fall into place before the door actually opens.
Brilliant analogy.
Yes.
And the availability of those specific tumblers, the co-regulatory proteins, varies drastically depending on the tissue.
Okay, so it's environment dependent.
Right. An estrogen receptor in breast tissue is surrounded by a completely different microenvironment of coactivators than an estrogen receptor up in the frontal lobe of the brain. The textbook illustrates this perfectly with the synthetic compound tibolone.
Tibolone. That's a masterclass in tissue specificity because it's structurally related to testosterone derivatives, but its clinical effects are remarkably compartmentalized.
Precisely. Because of the unique enzymatic environments in different tissues, tibolone is metabolized differentially. So in the brain and the skeletal system, its metabolites exhibit oestrogenic effects, and that can alleviate menopausal mood symptoms and prevent osteoporosis.
Right.
However, down in the breast and the endometrium, local enzyme systems convert it into metabolites that have progestogenic or weak androgenic effects.
Meaning it provides the neuroprotective and mood-enhancing benefits of estrogen centrally, without stimulating the aggressive cellular proliferation in the breast or uterus that, carries a high risk of malignancy with traditional unopposed estrogen therapies.
Exactly. It exemplifies the development of SERMs, or selective estrogen receptor modulators. By exploiting that tissue specificity, we can theoretically design psychiatric interventions that target mood and cognition pathways without completely derailing the patient's broader endocrine health.
Right. Okay, so having established the structural rules, we need to actually examine the machinery. How do these hormones alter the brain to change behavior?
The mechanisms of action.
Right. And the textbook divides this into two distinct temporal pathways, genomic and non-genomic action. And understanding the difference here is so critical for understanding the timeline of psychiatric symptoms and, the delayed efficacy of our treatments.
Yeah, you have to grasp the timelines. So genomic action is the classical slow pathway.
Okay.
The lipophilic steroid diffuses into the cell, binds to an intracellular receptor, and the entire hormone receptor complex translocates straight into the nucleus.
Right.
There, it physically binds to specific DNA sequences known as hormone response elements, which are located in the promoter regions of target genes.
So it is actively dictating transcription.
Exactly. It recruits RNA polymerase, creates messenger RNA, and directs the ribosome to synthesize entirely new proteins. It literally alters the structural architecture and the receptor density of the neuron.
But this profound structural remodeling, it takes hours or even days to manifest clinically.
And it requires immense cellular energy.
Right. So to use an administrative analogy, genomic action is like a corporation's CEO drafting a major strategic memo.
I like that.
The memo has to be written, it's reviewed by legal, disseminated to department heads, and then the staff must physically change their daily workflows. It is a powerful structural shift, but it simply cannot happen instantly.
No, it's a slow burn.
However, if an organism is faced with an immediate threat, it cannot wait 6 hours for a new protein to be synthesized before initiating a fight or flight response.
No, you'd be eaten by the lion by then.
Ah.
Which necessitates the second pathway, non-genomic action.
Right.
This pathway bypasses the nucleus entirely. The hormone binds to receptors on the external cell membrane, or intercalates directly into the phospholipid bilayer.
And what does that do?
It instantly alters the fluidity of the membrane and changes the kinetics of ion-gated neurotransmitter channels.
So it is the frantic, company-wide Slack message demanding an immediate halt to production.
Exactly. The effect occurs within seconds to minutes. And the textbook uses this brilliant example of corticosteroid stimulation of aggressive behavior in rats to demonstrate how these two timelines integrate perfectly.
The rat fight study?
Yeah. So when two male rats are placed in a cage together, there is an immediate, massive surge in glucocorticoids in response to the social stress.
Right.
Within seconds, you observe the rats engaging in aggressive posturing and rapid attacks. Now, this immediate phase is entirely mediated by non-genomic action at the membrane level.
The Slack message fires, threat detected, posture aggressively immediately.
Yes. But the effect of that membrane-level action is fleeting. It fades rapidly.
Right, it can't last.
However, that rapid burst serves a crucial preparatory function. It buys the animal time. While the rat is engaged in the initial non-genomic skirmish, the slower genomic mechanisms have been triggered in the background.
The CEO's memo is working its way through the cell.
Exactly. So hours later, the genomic pathway has synthesized the structural proteins required for sustained energy metabolism and prolonged territorial defense.
And the researchers proved this elegant duality, didn't they? By administering a protein synthesis inhibitor to the rats.
They did.
When they blocked the genomic pathway from creating new proteins, the rats still exhibited the immediate, rapid, aggressive posturing, which proved the non-genomic pathway was perfectly intact.
Right.
but they could not sustain the aggression in the later phases.
Exactly. So a single hormone initiates a rapid behavioral reflex while simultaneously building the biochemical infrastructure required for a sustained behavioral state.
Wow.
When we evaluate a patient in an acute crisis versus chronic anxiety, we are literally observing the interplay of both these temporal pathways.
That's so illuminating. And this brings us to what I consider, honestly, the most profound section of the chapter, which is epigenetics and developmental psychoneuroendocrinology.
Oh, this is fascinating stuff.
Because it turns out that the co-authored book of Our Emotional Lives isn't just written in the present moment. Our early developmental experiences can fundamentally change the font, the margins, and the binding of the book itself.
Yeah, we are transitioning here from activational effects, hormones triggering a response in the moment, to organizational effects.
Organizational.
Right. Exposure to specific hormonal environments during critical developmental windows physically alters brain morphology and receptor density.
And the concept of epigenetic transmission, it just completely upends the traditional dichotomy of nature versus nurture. We are not just talking about psychological learning here.
No, we're talking about experience altering how the genome is read. Epigenetics involves biochemical modifications, such as DNA methylation or histone acetylation.
Right.
Now, these don't change the underlying genetic sequence, but they dictate whether a gene is heavily transcribed or entirely silenced.
Think of it as a physical bookmark placed on the DNA strand.
Exactly. And the textbook highlights this landmark study involving cross-fostered rats that vividly demonstrates this.
Oh, I loved this study.
Right. So researchers observed two types of rat mothers, those who engaged in high levels of pup licking and grooming, and those who were, well, neglectful.
So the maternal behavior itself was the environmental variable.
Yes. The researchers then took pups born to highly attentive mothers and had them raised by neglectful mothers, and vice versa.
The old switcheroo.
Exactly. And what emerged was just extraordinary. The maternal behavior of the adult offspring matched the behavior of their adoptive mothers, not their biological parents.
Right.
A pup born to a neglectful mother but raised by a highly attentive mother grew up to be a frailly attentive, high-grooming mother itself.
And this wasn't merely a learned social behavior, right? It was physically embedded into their neurobiology.
Completely. The intense tactile stimulation from the attentive adoptive mother caused a physical cascade that literally removed methyl groups from the promoter region of the glucocorticoid receptor gene in the pup's hippocampus.
So it unsilenced the gene.
Precisely. Removing that methyl group allowed transcription factors like nerve growth factor to access the DNA. As a result, the pug's brain synthesized a higher density of glucocorticoid receptors. And with more receptors, the adult rat's brain is highly sensitive to circulating cortisol, meaning it can shut off its stress response very quickly and efficiently.
Because it detects it faster.
Right. It becomes a physiologically calmer, more resilient animal.
So the early tactile environment literally rewrote the sensitivity of the pup's endocrine system.
Yeah.
For a clinician, the implications of that are staggering. Because when we treat a patient with a history of early childhood adversity, we are not just addressing psychological trauma.
No, we are likely dealing with an HPA axis that was epigenetically programmed for hyperreactivity during a critical developmental window.
Right.
Furthermore, this endocrine system continues to learn and adapt throughout adulthood, right, through mechanisms of habituation and sensitization.
Because hormones are rarely released in a steady, continuous stream. Their release is pulsatile.
Exactly. And the amplitude, frequency, and duration of those pulses carry crucial biological information.
So if an individual is repeatedly exposed to a predictable, moderate stressor, the system may adapt by decreasing the amplitude of the cortisol pulse. It essentially learns to ignore the false alarm. That's habituation.
Spot on. Conversely, if the stressor is severe or unpredictable, the system may undergo sensitization, where the endocrine response becomes exaggerated and hyperreactive to even minor subsequent triggers.
Right, the alarm gets more sensitive.
Yeah. And if a highly habituated system is suddenly exposed to a novel, extreme stressor, it can undergo dishabituation, rapidly ramping back up to full alert.
And the exact trajectory of that adaptation, whether a patient habituates or sensitizes, is heavily influenced by their baseline genetics, isn't it?
Oh, massively.
This is where the field of pharmacogenomics is actively altering our prescribing practices. Because we all share the same fundamental genetic blueprint, but we harbor these microscopic variations known as single nucleotide polymorphisms.
Right, the SNPs. These genetic polymorphisms dictate individual variation in enzyme efficiency and receptor sensitivity.
Right.
The text notes that specific polymorphisms in the glucocorticoid receptor gene or the corticotropin-releasing hormone receptor genes can profoundly alter a patient's response to psychiatric medications.
Which provides a totally biological rationale for why a patient might fail an adequate trial of a first-line SSRI. It may not be an incorrect diagnosis or poor compliance.
No, it could just be that their specific genetic polymorphism regulating glucocorticoid sensitivity renders that particular pharmacological mechanism completely ineffective.
Wow. It points towards a future where psychiatric treatment isn't based solely on a phenomenological checklist, but is guided by concrete biological markers.
Yes, we are steadily moving towards subtyping mood disorders based on the patient's unique genetic and endocrine profile.
Okay, so having explored the microscopic machinery, the genomic delays, the epigenetic bookmarks, the receptor polymorphisms, we must pull the lens back.
Look at the macro.
Right. We need to examine the macro systems that actually utilize this machinery to mediate our interaction with the world. And we begin with the body's primary alarm system, the hypothalamic pituitary adrenal axis.
The famous HPA axis. It occupies the absolute center of psychoneuroendocrine research. Every psychiatrist must understand its architecture intimately.
It is the physiological translator of psychological stress.
Quite right.
So let's map the cascade for the listener. A stressor, whether it is a physical injury or just the psychological perception of an impending threat, is registered by the brain, specifically routed through the amygdala to the hypothalamus.
Right. The paraventricular nucleus of the hypothalamus is the starting point. It synthesizes and releases corticotropin-releasing hormone, or CRH.
CRH, right.
This peptide travels down a highly specialized vascular network, the hypophyseal portal system, directly into the anterior pituitary gland.
Okay, and in the anterior pituitary, CRH binds to its receptors and triggers the cleavage of a massive precursor protein called pro-opiomelanocortin, or POMC.
POMC.
And that releases the second major domino adrenocorticotropic hormone, ACTH.
Exactly. Now, ACTH enters the systemic circulation and travels down to the adrenal glands, which sit right atop the kidneys. Specifically, it targets the zona fasciculata of the adrenal cortex.
And that stimulates the de novo synthesis and rapid release of the primary effector cortisol.
Right, cortisol. And cortisol orchestrates a massive systemic mobilization of resources.
Right.
It triggers gluconeogenesis in the liver to flood the bloodstream with glucose for immediate energy. It enhances cardiovascular tone, shunts blood away from the digestive tract towards skeletal muscle.
You don't need to digest lunch if you're running for your life.
Exactly. And it actively suppresses the immune system to prevent runaway inflammation from potential injury.
Now, in the acute phase, this massive mobilization is highly adaptive. It ensures survival. But the true elegance of the HPA axis lies in its negative feedback loop, right? The system has to know when to turn itself off.
It has to, yeah.
As cortisol levels rise in the blood, the steroid crosses back over the blood-brain barrier and binds to receptors in the hippocampus, the hypothalamus, and the pituitary gland.
The cortisol acts as a biological signal saying, the threat is being managed, cease the production of CRH and ACTH.
Okay.
And this precise feedback regulation relies on a delicate balance between two specific types of intracellular receptors in the brain. The mineralocorticoid receptor, or MR, and the glucocorticoid receptor, or GR.
MR and GR.
Right. Understanding the different kinetics of these two receptors is absolutely essential. So the mineralocorticoid receptors, the MRs, have a very high affinity for cortisol.
Meaning they bind it very easily.
Right, even at very low concentrations. However, they have a low total capacity. They become saturated very quickly.
Okay, so because of this high affinity, under normal, resting, non-stressed conditions, the low baseline levels of circulating cortisol are primarily bound to the missers.
Yes.
The MNRs are responsible for maintaining the everyday circadian tone of the system. They are the thermostat.
The thermostat, exactly. But when a major stressor hits and cortisol levels spike dramatically, the MRs are already completely saturated.
Right.
The excess cortisol then spills over and binds to the glucocorticoid receptors, the GRs.
And the GRs have a low affinity for cortisol, so they don't bind it easily, but they have a massive capacity.
Yes.
They are essentially the fire alarms. They're only fully activated during a major stress response.
Exactly. The activation of the GRs is what mediates the intense acute effects of stress. And crucially, it is the GR activation that eventually forces the negative feedback loop to shut the entire HPA axis down once the stressor passes.
Right.
The system shifts from an MR-dominant resting state to a GR-dominant stress state and
Yeah.
then back again.
But what occurs when the stressor does not pass? What happens in cases of chronic trauma, poverty, or, sustained domestic abuse? The fire alarm basically never turns off.
And this creates a state of allostatic load?
Allostatic.
Yeah. The system becomes exhausted by its own perpetual activation. The negative feedback loop fails, leading to sustained hypercortisolemia. And chronic exposure to high cortisol is profoundly toxic to the organism.
The patient begins to develop central adiposity, severe insulin resistance, and hyperglycemia, hypertension, and a compromised immune system.
Yeah, the physical toll is massive.
But the psychiatric manifestations are equally devastating, right? The chapter highlights two classic endocrine pathologies that represent the extreme poles of HPA dysfunction: Cushing's syndrome and Addison's disease.
Right. Cushing's syndrome is the clinical manifestation of massive, sustained cortisol excess, often due to a pituitary adenoma secreting ACTH.
Right.
And the psychiatric morbidity in Cushing's is staggering. Over 50% of these patients suffer from severe atypical depression. Furthermore, roughly 10 to 15% develop frank psychosis, characterized by paranoia, auditory hallucinations, and intense suicidal ideation.
From the cortisol.
Just from the cortisol.
Wow. And on the opposite end of the spectrum, we have Addison's disease, or primary adrenal insufficiency. The adrenal cortex fails, and the body cannot produce adequate cortisol.
Right. And in Addison's, the clinical picture is dominated by profound, crushing apathy, overwhelming fatigue, social withdrawal, and psychomotor retardation.
So it's incredibly easy to misdiagnose as a severe, treatment-resistant, melancholic depression.
Very common, yeah.
But the defining clinical revelation is that if you administer exogenous glucocorticoids to correct the hardware deficiency, the psychiatric software symptoms completely resolve.
Almost like magic, yeah.
Which highlights a mechanism that strikes me as a profound biological vulnerability. We established earlier that the hippocampus is densely populated with glucocorticoid receptors, right? And it is crucial for signaling the negative feedback loop to halt cortisol production.
Yes, the hippocampus puts the brakes on the HPA axis.
However, the text clearly details that sustained high concentrations of cortisol are actually neurotoxic to the hippocampus.
Highly neurotoxic.
Chronic hypercortisolemia inhibits brain-derived neurotrophic factor, BDNF. It causes the retraction of dendritic arborization and ultimately leads to enzymatic cell death and the physical atrophy of hippocampal tissue.
It's devastating.
If the organ responsible for shutting off cortisol is destroyed by cortisol, isn't the body designed to spiral into inevitable failure under chronic stress?
It seems like a design flaw, doesn't it?
Right.
But that vulnerability is exactly what underpins the chronicity of severe psychiatric disorders like treatment-resistant depression and post-traumatic stress disorder.
Right.
It is a catastrophic vicious cycle. Chronic stress elevates cortisol. Elevated cortisol induces glutamate excitotoxicity and destroys hippocampal neurons.
Okay.
The damaged hippocampus then loses its ability to send the negative feedback signal to the hypothalamus. Without that inhibition, cortisol levels remain chronically elevated, causing even further neurotoxic damage to the remaining hippocampal tissue.
Which perfectly explains why, on a functional MRI, we consistently observe significant volume loss in the hippocampus of patients with chronic PTSD or severe, long-standing depressive disorders.
Yet, the physical hardware is literally eroded.
But the clinical picture is not entirely hopeless, is it? Because the brain retains a degree of neuroplasticity.
Thank goodness, yes. We know that treating major depression with selective serotonin reuptake inhibitors, SSRIs, is not just about increasing serotonin in the synapse.
Right.
Over weeks, SSRIs actually increase the expression of BDNF, promoting neurogenesis and eventually restoring hippocampal volume and reversing the cognitive deficits.
Additionally, the text notes the use of medications like mifepristone, which is a potent glucocorticoid receptor antagonist.
Right.
In cases of severe psychotic depression, blocking the GRs can halt the neurotoxic effects of excess cortisol, restoring synaptic plasticity in the cortex and rapidly alleviating the psychotic symptoms.
We are directly intervening in the psychoneuroendocrine loop to halt structural brain damage.
Incredible. Now, that principle of hormonal influence on brain architecture extends beyond the stress axis. We must examine the axis of state and reproduction, the hypothalamic pituitary gonadal axis, or HPG axis.
The sex steroids.
Right. The sex steroids, estrogen, progesterone, and testosterone, they are profoundly active neuroactive compounds.
Very much so. Let us begin with estrogen. Estrogen is fundamentally a neuroprotective and mood-enhancing hormone. It heavily innervates the limbic system and directly modulates classical neurotransmitter pathways.
Right, estrogen upregulates the synthesis of dopamine and decreases its degradation. But its interaction with serotonin is perhaps the most clinically relevant.
Yeah, absolutely. Estrogen increases the density of serotonin receptors and alters presynaptic serotonin reuptake. In fact, a chronic hypoestrogenic state, such as during the perimenopausal transition, can render the serotonin system so sluggish that a patient may fail to respond to an SSRI altogether.
Because the serotonin receptors physically require estrogen to function optimally. This physiological reality demands we completely re-examine premenstrual dysphoric disorder, or PMDD. Because historically, the medical community frequently dismissed PMDD as a purely psychological complaint, or simply assumed it was caused by, abnormal hormone levels.
Yeah, and extensive research has totally disproved the abnormal level theory. Women suffering from PMDD generally possess completely normal absolute levels of circulating estrogen and progesterone throughout their cycle.
Okay.
The pathology is not in the baseline concentration. The pathology lies in the dynamics of hormonal withdrawal.
It is the abrupt drop that causes the catastrophe.
Exactly.
During the luteal phase of the menstrual cycle, following ovulation, levels of progesterone are high. Progesterone itself can sometimes have dysphoric effects, but its primary metabolite, allopregnanolone, is crucial.
Allopregnanolone is a highly potent neurosteroid. It binds as a positive allosteric modulator to the GABA-I receptor in the brain.
GABA, right.
Yeah. Its pharmacological action is remarkably similar to a benzodiazepine or alcohol. It produces profound anxiolytic, sedative, and calming effects.
Okay, so the brain acclimates to this high level of allopregnanolone. But just prior to menses, the corpus luteum degenerates and the production of progesterone and allopregnanolone plummets off a cliff.
It is a massive biochemical withdrawal. In vulnerable women, this precipitous decline in allopregnanolone triggers a structural plasticity event in the GABA-A receptor itself.
A structural change.
Yes. The rapid withdrawal increases the transcription of the alpha-4 subunit of the receptor.
And the structural shift renders the GABA receptor highly insensitive to normal modulation. It loses its ability to inhibit neural firing.
Exactly. The clinical result is a state of acute, internally generated neurochemical withdrawal. The brain's primary inhibitory system goes offline.
Wow.
Leading to the severe anxiety, extreme irritability, emotional lability, and profound depressive symptoms that characterize PMDD. And the moment menses begins and the hormonal landscape resets, the symptoms often rapidly dissipate.
It is an embodied withdrawal syndrome, and we observe a similar, though exponentially more massive, dynamic in the immediate postpartum period, right? Which explains the high incidence of postpartum depression and postpartum psychosis.
Spot on. Throughout the third trimester of pregnancy, the placenta acts as a massive endocrine organ.
A huge one.
Right. Flooding the maternal brain with unprecedented concentrations of estrogen, progesterone, and neurosteroids, like allopregnanolone. This creates a highly buffered, generally positive mood state and deeply suppresses the HPA stress axis.
Then the child is born, the placenta is delivered, and that massive endocrine factory is abruptly removed from the system.
The hormone levels crash to near zero within hours.
It is arguably the most dramatic, rapid endocrine shift a human body can endure. The sudden withdrawal of allopregnanolone leaves the GABAergic system entirely exposed and deeply vulnerable, creating the biological substrate for severe postpartum mood disorders.
Which is exactly why the development of brexanolone represents a paradigm shift in psychiatric treatment.
Right, brexanolone, because it's an FDA-approved synthetic proprietary formulation of allopregnanolone.
Exactly. So instead of giving a postpartum patient a standard SSRI and waiting 4 to 6 weeks for genomic neuroplasticity to occur, brexanolone is administered as a continuous intravenous infusion over 60 hours.
Wow.
It directly replaces the precise neurosteroid that was lost during placental delivery, immediately restoring the function of the GABA-A receptors. The rapid alleviation of severe depressive symptoms is an absolute triumph of targeted psychoneuroendocrinology.
Incredible. Now, shifting to the androgens, we have to dismantle the pervasive cultural caricature of testosterone because the popular narrative is that testosterone is simply a rage molecule, a blunt instrument that universally causes violence and aggression.
Yeah, that narrative is biologically illiterate.
Right.
Testosterone is a complex neurosteroid with extensive genomic and non-genomic actions across the brain. Now, while supraphysiologic doses in certain animal models can increase aggression in humans, the behavioral output is heavily modulated by the individual's baseline personality, their social context, and the existing hierarchy.
The textbook cites a brilliant ethological study involving cynomolgus monkeys that completely reframes how we should view testosterone.
Oh, this study is fantastic.
Okay.
So, in an established social hierarchy of monkeys, researchers administered exogenous testosterone.
Okay.
If the testosterone was given to a dominant, high-ranking monkey, that animal exhibited an increase in dominant assertive behaviors to maintain its rank.
Makes sense. However, when they administered the exact same dose of testosterone to a low-ranking submissive monkey, the animal did not suddenly attack the alpha.
No, not at all.
Instead, the exogenous testosterone amplified its submissive behaviors. It became more withdrawn and deferential.
Right. Because the hormone does not blindly create aggression. It amplifies the existing behavioral strategy required to maintain the animal's specific position within its social environment.
Wow.
In clinical practice, administering testosterone replacement to hypogonadal men frequently results in improved mood, increased vitality, and a decrease in irritability and dysphoria.
Right, context is everything. And alongside testosterone, we must examine dehydroepiandrosterone, or DHEA.
Ah, DHEA.
DHEA is the most abundant circulating steroid in the human body, produced largely by the adrenal glands, but it is also synthesized independently within the central nervous system as a neurosteroid.
Yeah, and DHEA serves as a crucial counter-regulatory molecule to cortisol.
The counterweight.
Exactly. While cortisol mediates the catabolic tissue-breaking aspects of the stress response, DHEA exerts powerful anti-glucocorticoid and neuroprotective effects. It promotes neurogenesis and protects the hippocampus from glutamate excitotoxicity.
And this antagonistic relationship introduces one of the most compelling concepts in the chapter for future clinical application, the resilience ratio.
Right. Specifically, the molar ratio of cortisol to DHEA.
Okay.
If you only measure a patient's cortisol level, you're only seeing the amount of damage being inflicted. You must also measure the DHEA to understand how much neuroprotective buffering the brain is receiving.
And the text highlights that patients suffering from chronic PTSD or severe major depressive disorder consistently present with an abnormally low cortisol to DHEA ratio.
Yeah. A low ratio indicates that the toxic effects of cortisol are essentially unopposed.
Right.
Conversely, individuals who undergo severe trauma but do not develop PTSD often demonstrate a robust, high ratio of DHEA to cortisol. It is a quantifiable biological index of an individual's innate physiological resilience to stress.
That is amazing. That principle of endocrine imbalance dictating psychiatric presentation brings us to the hypothalamic pituitary thyroid axis, the HPT axis.
Yeah.
I want to offer stark warning based on this section. When a patient presents to the emergency department with acute, bizarre, or treatment-resistant psychiatric symptoms, the clinician must maintain a high index of suspicion for clinical mimicry.
Oh, absolutely. The thyroid governs the basal metabolic rate of virtually every cell in the body, and the brain is exceptionally sensitive to fluctuations in T3 and T4.
Right.
Thyroid dysfunction can flawlessly masquerade as almost any primary psychiatric disorder.
Let's examine the extremes here. In hyperthyroidism, or thyrotoxicosis, the gland is producing an excess of thyroid hormone. The metabolic engine is running far too hot.
Right. Clinically, this presents with tachycardia, diaphoresis, heat intolerance, and a fine tremor. But the psychiatric phenotype often dominates the presentation.
What does that look like?
The patient exhibits profound anxiety, severe insomnia, emotional lability, and psychomotor agitation. It can look identical to a severe panic disorder or a mixed manic episode.
And in severe cases, known as thyroid storm, it can progress into a florid acute psychosis, frequently marked by intense paranoia and visual hallucinations.
Yeah.
If you admit that patient to an acute psychiatric ward and merely prescribe an antipsychotic, you are missing a potentially lethal metabolic crisis.
A huge mistake. Conversely, hypothyroidism, a deficiency in thyroid hormone, slows the metabolic engine.
Right.
The patient presents with cold intolerance, weight gain, bradycardia, and dry skin.
And psychiatrically, hypothyroidism is the great imitator of melancholic depression. The patient suffers from crushing fatigue, anhedonia, decreased libido, profound psychomotor retardation, and significant cognitive fog.
In the elderly, severe hypothyroidism can cause reversible cognitive impairment that perfectly mimics the onset of dementia. It was historically termed myxoedema madness.
Myxoedema madness. This is exactly why a basal thyroid stimulating hormone, or TSH level, is an absolute mandatory screening lab in any comprehensive psychiatric evaluation.
Absolutely. Treating the underlying thyroid dysfunction will frequently resolve the psychiatric symptoms entirely, rendering psychotropic medications completely unnecessary.
However, we also must be deeply aware of how our own primary psychiatric interventions can disrupt this delicate axis.
Yes, the iatrogenic effects.
Right. Lithium remains the gold standard mood stabilizer for bipolar disorder, but its side effect profile is an endocrine minefield.
It really is. Lithium possesses a unique elemental similarity to iodine, and it aggressively interferes with the thyroid gland at multiple stages.
How so?
Well, it concentrates within the thyroid and actively inhibits the iodination of tyrosine, blocks the release of mature T3 and T4 into the bloodstream, and impairs the gland's cellular response to TSH.
So the clinical consequence is that approximately 30% of patients maintained on long-term lithium therapy will develop an elevated TSH, and a significant percentage will progress to frank clinical hypothyroidism.
Yeah. Therefore, a patient who's been entirely stable on lithium for a decade, who suddenly presents with anergia and depressed mood, they're likely not experiencing a breakthrough depressive episode of their bipolar disorder.
Right.
They're likely suffering from lithium-induced hypothyroidism.
But lithium's interference doesn't stop at the thyroid. Its interaction with the parathyroid gland is arguably more dangerous and often missed.
Yes, the parathyroid glands manage the body's calcium homeostasis.
Okay.
Lithium directly alters the sensitivity of the calcium-sensing receptor, the KSR, on the chief cells of the parathyroid gland. It essentially raises the biological set point.
Meaning the gland requires a higher concentration of circulating calcium before it will shut off the secretion of parathyroid hormone, or PTH.
Exactly. This leads to a persistent state of hyperparathyroidism and resulting hypercalcemia. And excess calcium in the blood is highly neurotoxic.
Elevated calcium disrupts neurotransmitter release and voltage-gated ion channels. Mild hypercalcemia can cause lethargy and depressive symptoms, but acute or severe hypercalcemia can precipitate a rapid onset delirium.
Yeah.
Profound confusion and severe psychosis, classically characterized by vivid visual hallucinations.
And the textbook explicitly notes that long-term lithium administration can actually stimulate the cellular proliferation of parathyroid tissue, leading to the formation of a parathyroid adenoma.
A benign tumor.
Right, but it autonomously secretes massive amounts of hormone. If a patient on long-term lithium suddenly develops delirium and hallucinations, checking their serum calcium and intact PTH is an absolute medical emergency.
Because surgical removal of the adenoma is often curative for the psychiatric symptoms.
Exactly. It just underscores the reality that a psychiatrist cannot simply manage the mind. They must act as an astute internist and endocrinologist.
Absolutely. Before we move to the final integration, we need to spend time on what the chapter categorizes as the supporting cast of modulators.
The neuropeptides.
Right. These are peptides that exert profound psychiatric influence but are often overlooked. We begin with prolactin. Outside of obstetrics and lactation, prolactin is deeply intertwined with our dopaminergic systems.
Yes. Prolactin secretion from the anterior pituitary is under constant tonic inhibitory control by dopamine.
The dopamine brake.
Exactly. Dopamine travels down the tuberoinfundibular pathway and binds to D2 receptors on the lactotroph cells, strongly suppressing the release of prolactin.
And the clinical relevance emerges when we prescribe classical antipsychotic medications, which are fundamentally D2 receptor antagonists.
Right.
By blocking those dopamine receptors to treat schizophrenia or mania, we inadvertently sever the inhibitory brake on the pituitary.
The result is rapid and severe hyperprolactinemia. The pituitary floods the system with prolactin.
And physically, this causes galactorrhea, amenorrhea in women, gynecomastia in men, and long-term osteopenia. But the psychiatric effects are equally debilitating.
Very much so. High prolactin levels suppress the HPG axis, plummeting testosterone and estrogen, leading to profound sexual dysfunction, anhedonia, and a marked intolerance to stress.
So we are often attempting to treat the negative symptoms of schizophrenia, the apathy and withdrawal, while simultaneously prescribing a medication that induces a hormonal state that mimics and exacerbates those exact same symptoms.
It's a real clinical bind.
Yeah.
Following prolactin, we must examine the neurohypophyseal peptides oxytocin and vasopressin. These two ancient molecules are synthesized in the supraoptic and paraventricular nuclei of the hypothalamus and stored in the posterior pituitary. And they operate as a fascinating reciprocal pair.
Oxytocin is colloquially known as the cuddle hormone, which is honestly a gross oversimplification.
Very much so.
It does promote affiliative behavior, pair bonding, and maternal care. but I found its cognitive effects startling. The text refers to oxytocin as an amnestic peptide.
Yeah, that's wild. While it lowers anxiety by dampening amygdala reactivity, oxytocin remarkably impairs the consolidation and retrieval of memories.
Why would it do that?
Well, from an evolutionary perspective, it is hypothesized that this amnestic property helps maternal organisms forget the intense physical trauma of childbirth, facilitating immediate bonding with the infant.
Wow. But sitting directly opposite oxytocin on the biochemical seesaw is vasopressin, also known as antidiuretic hormone. If oxytocin is the peptide of bonding and forgetting, vasopressin is the peptide of stress and remembering.
Exactly. Vasopressin acts synergistically with CRH to powerfully amplify the HPA stress response. It heightens arousal, increases aggressive territorial behaviors in certain models, and critically, it strongly enhances the consolidation of aversive traumatic memories.
And the textbook highlights a bizarre clinical phenomenon directly linked to vasopressin dysregulation psychogenic polydipsia in schizophrenia.
Yeah, this is severe. Up to 15% of chronically hospitalized patients with schizophrenia develop polydipsia, an uncontrollable compulsive drive to drink massive quantities of water.
And this is not merely a psychological compulsion. It's frequently driven by an inappropriate central secretion of vasopressin.
Right. The excess vasopressin drives a pathological thirst mechanism and forces the kidneys to retain free water, rapidly leading to severe hyponatremia water intoxication.
Which can precipitate lethal seizures and coma?
Yeah, the psychological behavior is a manifestation of a peptide imbalance.
The final supporting peptide we must cover powerfully illustrates the gut-brain axis cholecystokinin, or CCK. Most physicians know CCK purely as a digestive hormone secreted by the duodenum to stimulate the gallbladder to release bile after a fatty meal.
The gallbladder hormone.
Yet it is astonishing to realize that CCK is also one of the most abundant neuropeptides in the central nervous system, heavily concentrated in the limbic system, the amygdala, and the cortex. It is deeply entangled with our anxiety circuits.
Very deeply. The central CCK system, primarily operating through the CCKB receptor, acts as a potent anxiogenic signal. It heavily modulates dopamine release in the ventral tegmental area.
And the evidence for its role in anxiety is unequivocal. The text details that if you administer an intravenous infusion of a specific fragment, CCK4, to a human being, it will reliably and predictably trigger a massive, severe panic attack.
It's quite literally a panic injection. It induces a full physiological and psychological panic cascade in healthy volunteers. But more critically, patients with a pre-existing diagnosis of panic disorder are exquisitely hypersensitive to CCK4. They will experience a florid panic attack at a microdose that would barely register in a healthy control subject.
This proves that panic disorder involves a hyperreactive, hypersensitive CCK receptor system. Panic is not simply a cognitive spiral of catastrophic thinking. It is a peptide-driven biological event.
Exactly. Exploring selective CCKB receptor antagonists offers a very promising non-benzodiazepine pharmacological target for severe anxiety disorders.
Okay, having navigated this vast, complex chemical factory, the ultimate challenge is integration. How does the modern clinician synthesize this knowledge when a patient actually sits in their consulting room?
The primary paradigm shift is the complete dissolution of the boundary between a psychological symptom and a physiological state. We can no longer dismiss profound fatigue, anhedonia, or panic as purely functional or psychogenic. They are embodied phenotypes of endocrine dysregulation. When a patient reports an inability to experience pleasure, we must evaluate the dopaminergic pathways, certainly, but we must simultaneously evaluate the allostatic glucocorticoid load that is actively suppressing that dopamine, or the hypothyroid state that is dampening the receptor sensitivity.
The psychiatric symptom is merely the final, common, observable pathway of a deeply interacting biochemical network.
Exactly.
Which raises a crucial question. If the science is this robust and the mechanisms are this clearly delineated, why isn't endocrinology a more explicit structural component of our diagnostic manuals like the DSM-5?
That's the big question.
Why aren't we formally subtyping major depressive disorder by cortisol levels or DHEA ratios today?
Well, the resistance is rooted in significant diagnostic and methodological challenges. The primary obstacle is differentiating state versus trait.
State versus trait, right.
If a patient presents with elevated cortisol during a severe depressive episode, is that hypercortisolemia a permanent biological trait that caused the depression? Or is it merely a transient physiological state reflecting the acute stress of the current illness?
Disentangling the chicken from the egg in a cross-sectional clinical snapshot is exceptionally difficult.
It really is. Furthermore, we are dealing with massive confounding variables.
Right.
The endocrine system is constantly shifting. A patient's hormone levels fluctuate wildly based on circadian rhythms, recent dietary intake, sleep architecture, phase of the menstrual cycle, age, and immediate environmental stress.
Establishing a clean, standardized baseline across a broad psychiatric population is statistically very noisy.
Extremely noisy. Additionally, we currently lack the massive longitudinal cost-effectiveness trials required to prove that running complex, expensive endocrine panels on every single psychiatric admission significantly improves long-term clinical outcomes compared to standard assessment.
So the field simply lacked the definitive, clean criteria to incorporate it into the current DSM.
Yes, for now.
However, the trajectory of the science is undeniable. The future of psychiatric diagnosis will inevitably rely on biomarkers and endophenotypes.
Whoa, without a doubt. We are moving toward a reality where we will utilize the dexamethasone CRH stimulation test to objectively probe the integrity of a patient's HPA negative feedback loop.
Right.
We will measure the DHEA to cortisol ratio to quantify their biological resilience. We will transition from a symptom cluster diagnosis like major depressive disorder to a biologically precise diagnosis, such as a depressive phenotype primarily driven by HPA axis hyperreactivity, epigenetic GR promoter methylation, and profound allopregnanolone withdrawal.
That level of precision will fundamentally revolutionize how we select and sequence treatments.
It is the ultimate synthesis of the hardware and the software.
We have covered an immense amount of complex physiology today. Let us consolidate the core truths, the key takeaways from this chapter that must inform your clinical reasoning.
Right. First, psychoneuroendocrinology establishes that the brain and the endocrine system operate in continuous bidirectional feedback loops. The brain is an active endocrine organ, and the classical endocrine glands are direct neural effectors.
Second, the hypothalamic pituitary adrenal, or HPA axis, is the central translator of perceived psychological stress into physiological reality. While acutely adaptive, chronic activation results in allostatic load, causing neurotoxic damage, specifically structural atrophy in the hippocampus, driving the pathology of trauma and mood disorders.
Brilliant. Third, endocrine effects operate on multiple simultaneous timelines. Hormones utilize rapid non-genomic action at cell membranes for immediate behavioral responses, while simultaneously employing slower genomic action to alter DNA transcription for sustained structural neural changes.
Fourth, profound psychiatric symptoms are frequently the direct manifestation of endocrine dysregulation, whether it is the acute panic of PMDD driven by allopregnanolone withdrawal, the severe psychosis resulting from a lithium-induced parathyroid adenoma, or the energic depression of undiagnosed hypothyroidism. The chemistry dictates the state.
And finally, our therapeutic interventions must account for this embodied reality. A patient with treatment-resistant depression may not need a fifth trial of an SSRI. They may require thyroid optimization.
Right.
An anxious patient may be suffering from the physiological exhaustion of an unbuffered HPA axis. We must treat the entire integrated system rather than suppressing isolated symptoms.
Excellent. Now, to solidify these concepts, we'll conclude with a clinical reasoning exercise. I will present a series of brief clinical vignettes and questions. I will pause to allow you, the listener, to formulate your differential, and then our expert here will break down the precise mechanism and the correct reasoning. Think of this as a peer review of complex cases.
That's good.
Question 1. You observe a laboratory model where a rodent is exposed to a sudden novel threat. Within seconds, the animal exhibits a complex evasive behavior. Given the rapidity of this response, which specific hormonal mechanism must be mediating this behavioral change, as it occurs far too quickly for gene transcription?
Well, give yourself a second to think, right? The mechanism is non-genomic action. This occurs when steroid or peptide hormones bind directly to surface membrane receptors or intercalate into the phospholipid bilayer, instantly altering ion channel kinetics and neuronal excitability. It bypasses the nucleus entirely, serving as the immediate, rapid response system of the endocrine network.
Spot on. Question 2. A 45-year-old patient with a long history of severe chronic post-traumatic stress disorder undergoes a functional MRI for a research study. The imaging reveals a significant bilateral reduction in hippocampal volume. What specific endocrine mechanism is the primary driver of the structural neurotoxicity and cellular atrophy?
Have a think about the feedback loop. The structural damage is driven by chronic hypercortisolemia, leading to enzymatic cell death. In chronic stress states, the HPA axis becomes dysregulated, flooding the brain with cortisol. Sustained high levels of cortisol suppress BDNF, cause dendritic retraction, and induce glutamate excitotoxicity, specifically in the hippocampus. This creates a dangerous positive feedback loop, as the damaged hippocampus can no longer effectively inhibit the HPA axis.
Perfect. Question 3. A 30-year-old woman presents to the clinic reporting severe, debilitating episodes of anxiety, marked irritability, and suicidal ideation that occur exclusively in the 5 days preceding her menses, resolving completely on day 2 of her cycle. Current research into PMDD indicates that these symptoms are not caused by abnormal absolute hormone levels, but rather by a specific structural change in a neuroreceptor. What is the precise mechanism?
Think about the withdrawal state here. The mechanism is the altered structural sensitivity of the GABA receptor due to the rapid withdrawal of allopregnanolone. During the late luteal phase, the precipitous drop in this calming neurosteroid triggers a plastic change in the GABA receptor, specifically an increase in the alpha-4 subunit. This renders the receptor insensitive to normal inhibitory modulation, effectively throwing the brain into a state of acute internal neurochemical withdrawal.
Excellent. Question 4. A 55-year-old patient with bipolar type 1 disorder who has been clinically stable on lithium monotherapy for 12 years is brought to the emergency department by their family. They present with sudden onset delirium, profound lethargy, and vivid visual hallucinations of animals in the room. Beyond checking a lithium level, what specific endocrine system must be investigated immediately due to the long-term effects of this medication?
Remember the other glands in the neck. You must immediately investigate the parathyroid gland and check serum calcium and intact parathyroid hormone levels. Long-term lithium exposure alters the calcium-sensing receptor threshold and can stimulate the growth of a parathyroid adenoma. This results in hyperparathyroidism and severe hypercalcemia, which acts as a neurotoxin and classically precipitates acute delirium and visual hallucinations.
Brilliant. Final question number 5. You are assessing a patient who recently survived a severe, life-threatening accident. In attempting to evaluate their physiological resilience and their biological risk for developing persistent chronic PTSD, researchers often look beyond isolated cortisol levels. What specific hormonal ratio provides a more accurate index of how effectively the individual's brain is buffered against the neurotoxic effects of the stress response?
Think about the counterweight to cortisol. The key metric is the ratio cortisol to dehydroepiandrosterone, or DHEA. While cortisol mediates the catabolic damage of the stress response, DHEA is a neurosteroid that exerts powerful anti-glucocorticoid and neuroprotective effects. A low cortisol to DHEA ratio indicates robust biological buffering and is generally predictive of higher physiological resilience to trauma.
And that concludes our exploration of the endocrine mind. I want to leave you with a final thought to mull over before your next clinical encounter. If the sudden withdrawal of a single neurosteroid like allopregnanolone can plunge a mind into a suicidal abyss, if a microscopic infusion of a gut peptide like CCK4 can induce sheer unadulterated panic, and if the simple act of maternal grooming can physically strip methyl groups off DNA to permanently rewire an infant's stress response, where exactly does the concept of an individual personality end and the reality of complex chemistry begin?
That's a huge question.
Are our emotional lives merely the sum of our hormone receptors, desperately fighting to maintain homeostasis in a chaotic environment? It is a philosophical provocation, but one that fundamentally alters the empathy and precision we bring to patient care.
We strongly advise you to use this discussion as a companion guide and read directly alongside Chapter 1.12 in Kaplan-Sadok's comprehensive textbook of psychiatry. Engaging with the primary text will solidify your grasp of these mechanisms and elevate your clinical reasoning.
Here are the details of this episode.
Psychoneuroendocrinology. Byline: The Chemistry of Stress and State, How Brain and Body Co-Author Emotional Life.